All right. So membranes are made of phospholipids. So again, phospholipids are very critical for cells to be viable. You need phospholipids because you need membranes. And membranes, of course, a very important structural element of cells. Allows for a protected environment. So the membrane basically creates a barrier, and within that barrier, inside of the cell, we can control things like concentrations of nutrients that we need for energy, synthesis of proteins, control of pH, control of other aspects of the cell, the contents, the concentration of ions, all that good stuff. We are able to do that because membranes form a barrier that polar and charged particles cannot pass through easily and or at all, pretty much, in terms of physiologically. So again, it allows cells to be able to collect nutrients, also provides a hydrophobic area in the membrane itself that the cell can use for things like the electron transport chain, which we use to make ATP in mitochondria. Prokaryotes only have the one membrane, the outer cellular membrane. They don't have any membrane-bound organelles, so prokaryotes, again, simpler model of cell, and just have that single membrane. And so basically everything they need a membrane for, they have to use that membrane for. So unlike ourselves, we do oxidative phosphorylation using the electron transport chain in mitochondria. Prokaryotes just use their cellular membrane to do that kind of stuff. Eukaryotes, of course, multiple membrane-bound organelles, including the nucleus, which is actually a double membrane, as is the mitochondria, the ER and Golgi that basically interact with each other, and are very important for protein packaging. Peroxisomes and lysosomes, basically the little stomachs of your cell, contain digestive enzymes and low pH, and in the case of peroxisomes, we have oxygen species, in order to break down the things we take into our cells. Note that each membrane in a eukaryotic cell is going to have a different composition of phospholipids. Over here on the left, the top 6 items here are all types of phospholipids. Phosphatidylcholine, phosphoethanolamine, sphingolipids, phosphatidylinositol, serine, cardiolipin. All of these are different types of phospholipids, and you can see that each of the membranes here, nuclear membrane, plasma membrane, lysosomal membrane, mitochondrial membrane, Golgi membrane, have different compositions. Most of them contain a lot of phosphatidylcholine. We can see the nuclear membrane contains the most, and the plasma membrane contains the least. You can also see cholesterol here in green. You can see the amount of cholesterol varies. The more cholesterol that's found in a membrane, the more fluid it is. When we talk about the fluid mosaic model of the membrane, you'll see why membrane fluidity actually is pretty important for some membranes. Even the mitochondria, even though it has a very small amount of cholesterol, it has a huge amount of protein in it compared to these other membranes. But it's part of its overall functionality, and so it can get by with less cholesterol than the other membranes. So again, you don't need to memorize the specific values here or anything like that. Just understand that levels, the amounts of the different types of phospholipids varies depending on which membrane you look at. And I would like you to know that the mitochondria has the least amount of cholesterol out of all of these different membranes. I would like you to know that. Other than that, just be aware that they vary. They vary in the amount of phospholipids, and they vary in the amount of cholesterol. So membrane structure, the idea of the biological membrane is similar to just the concept of a micelle. And a micelle is made from anything that has an amphipathic nature, like detergents, for instance, where you have basically a polar or charged head group and then a hydrophobic tail. When you do this and you take these types of molecules and you put them into an aqueous environment, the first thing they're going to do is form these micelles. And what this is the hydrophobic part of the amphipathic particle trying to avoid water. So it basically comes together with a bunch more of these particles and forms what we call a little micelle, like you see here. Micelles are what form when we use soap, when we use detergent. It's the same idea. You have amphipathic molecules that you want to, we call emulsify. This is why when you use soap or detergent, you have to scrub in order for to remove dirt and oil and grime. And that's because you're basically taking these particles and you're mixing them with the oil. Then, once you rinse and you get rid of the soapy stuff, basically they're forming these micelles. And the oil and stuff gets trapped inside of these micelles in the hydrophobic part. That's what you do when you use detergent to clean clothes, when you use soap to clean your hands. That's what leads to them being able to clean stuff. With phospholipids, when you first start adding them to an aqueous solution, first you're going to get micelles like you see here. But if you keep adding them to solution, they will spontaneously form membranes like this, where you actually get two sides to the layer of phospholipids. And this is when you get the phospholipid bilayer. So when you get enough phospholipids in an aqueous environment, if you keep adding them, they'll start to form these vesicles. And what's amazing is they do this spontaneously. Again, they're trying to keep the fatty acid tails away from water, and so they tend to congregate and point towards each other. And eventually, if you add enough, you start to get this inner layer along with the outer layer. So it goes from a micelle to a vesicle. And if you think about it, a cell is just a really, really big vesicle. And again, that leads to what we call the phospholipid bilayer, where we have two layers of phospholipids, one facing the polar head groups are facing out on one side, the polar head groups are facing in. In between, you have your fatty acids that are going to associate with each other to form the bilayer. The inner portion of this bilayer, the yellow part, is going to be hydrophobic. And guess what? Once again, particles that have charges or are polar are unable to pass through the bilayer. Again, they form spontaneously. There's a concentration that's called the critical micelle concentration, and you know that if you reach that concentration, you're going to get formation of micelles. And eventually, they will start to form bilayers from the phospholipids if you keep adding them, and eventually you start getting those bilayers. So again, this happens spontaneously. What's really cool about this as well is, again, all of these particles are just associated with each other through weak force interactions, in particular hydrophobic interactions, but they don't have any covalent bonds between them. Okay? That's pretty amazing, because what that means is we can put stuff into this membrane, we can manipulate it, we can do stuff with it, and we don't have to worry about breaking it or causing damage to it, because these particles just kind of stay next to each other to form these barriers. We don't have to covalently link them. And again, to avoid water, the vesicles end up forming, and eventually you get your double bilayer of phospholipids, and that is what ends up forming our membranes. If you just have a vesicle that just has the phospholipids in it, nothing else, we call those, just as a generic term, those are called liposomes. Very stable, and again, the basis of biological membranes. If you just put these into an aqueous environment, they're going to sit there and form these vesicles, and they're going to be very happy to do it. They'll just stay in that form, unless you come in and you mess with them, either by adding detergents or emulsifying them. But again, they'll just spontaneously go back to forming these structures, even if you do that. And the barrier prevents polar and charged molecules from passing. Fat-soluble stuff can pass through, but remember, fat-soluble stuff generally doesn't just float around in an aqueous environment. So really, we need mostly to prevent the polar and charged particles from being able to pass through freely. All right, so that's vesicles. Now we'll move on to the actual plasma membrane.